Biolog's technology platforms provide an important new tool for the characterization and development of cell lines. To gain full value of this slideshow, first-time viewers are encouraged to watch the Technology Overview slide set found at the beginning of this series. As noted throughout the slideshows in this series, Biolog's platforms are versatile in serving multiple application areas. While this slideshow emphasizes applications in cell line characterization and development, Biolog's platforms are being applied to bioprocess development, studies in mitochondrial function and chemical toxicology, cancer research, in metabolism and metabolic disorder studies, and in genotype-phenotype studies. Biolog's platforms are based on an important biological observation. Each cell type or cell line is unique in its overall profile of energy substrate utilization and chemical sensitivity. This selectivity allows Biolog's platforms to simply and inexpensively evaluate different or altered cells with a very high level of discrimination. This selectivity can also be readily applied to identifying appropriate clones, leading to the selection of cell lines with preferred phenotypic profiles. Biolog offers a variety of test panels, and some or all of these can be used in phenotypic testing of cell lines. The assays examine stimulation or inhibition of cells by carbon and nitrogen energy substrates, ions, hormones and metabolic effectors, anti-cancer agents. A toxicology assay is also available using 8 energy substrates. More information on the use of this assay is presented in the toxicology slide set in this series. When a panel of substrates and chemical agents are utilized in comparison studies, the power of Biolog's platforms to easily characterize and discriminate cell types is readily apparent, as in this example with C3a liver cells and A549 lung cells. With Biolog's OmniLog instrument and software, one can record and compare kinetic cell phenotypes. In the example shown here, the A549 cell's metabolic response is recorded in green, the CHO cells in red, and yellow indicates the overlap where the cells are responding the same. Clearly, these cell lines have very different phenotypic properties. Biolog's technology can be applied even to distinguishing clones derived from parental cells. Here, the C3a subclone line is compared to its parental cell line, HepG2. The 4 plates used for each cell line contain the full complement of 367 carbon substrates. While the results are indeed very similar, the C3a clone indicates a stronger metabolism of pyruvate. In fact, the C3a subline was obtained by selecting for a clone that could grow better on pyruvate. Clear metabolic differences can also be seen between primary cells and immortalized cells. Here, we show data showing substantial differences between primary rat hepatocytes and the immortalized C3a liver cell line. As noted in earlier slides, cells also display unique chemical sensitivity phenotypes. The following 2 slides illustrate the power of Biolog's platforms to capture and profile these particular phenotypes. Here is a chemical sensitivity comparison of a mouse embryo fibroblast cell line compared with its RAS-activated isogenic line. The chemical sensitivity assays detect an increase in resistance to 17 ions and anti-cancer agents in the RAS-activated cells. Assay of another isogenic pair of cell lines obtained from ATCC confirms that the DX5 subline is doxorubicin and daunorubicin resistant, but have identical sensitivities to methotrexate. The next 3 slides profile the important role Biolog's platform provides in characterizing cell stability and aging, and in QC applications. Biolog's PM assays are easy to run and highly reproducible. This is critical in establishing a consistent phenotypic baseline for monitoring any changes in stability and aging, and in confirming cell identity. In this dendrogram, based on Biolog's platform results, the cell lines evaluated for their metabolic phenotypes are clustered. The cluster groups correlate with their tissue of origin, starting with two liver lines, the A549 lung line, followed by prostate and colon cell lines. Within the A549 cell cluster, we can examine further the effects of multiple passages. As noted here, the A549 cells display substantial stability from passage 17 to 52, but by passage 54 there is a substantial shift detected in the metabolic phenotype. Although many cell lines will be metabolically stable when maintained on the same culture medium, it is also possible to alter the metabolic properties of cells in culture. In this instance, the liver cell line HepG2 underwent a series of selective media adjustments to bring its metabolic phenotype more closely toward that of a normal hepatocyte. Many carbon energy pathways have been successfully activated. Cells undergoing differentiation display phenotypic shifts in phenotypes, including substrate preferences. Biolog's platform can be used to reveal these shifts. This has particular application in the investigation of stem or progenitor cells, where various energy substrates or other chemicals can potentially arrest or promote differentiation, or even give rise to dedifferentiation. In all these particular situations, Biolog's platform can be used to help assess and determine these valuable cellular responses. In a study conducted at the Broad Institute at MIT, Biolog's assays were used to detect and define shifts in substrate metabolic rates as fetal preadipocyte white or brown fat cells differentiated to become adult cells. PM technology is a fundamentally important new tool for metabolic profiling of cells. It can identify preferred substrates and energy pathways. It provides cell identification. It can also be a sensitive metabolic monitor for cell stability and aging. The platform correlates substrates and cell survivability, as well as correlate substrates and cell differentiation. Cell metabolism lies at the core of many key application areas. Understanding this dimension of cellular behavior has important research and commercial value. Biolog's metabolic profiling platforms stand alone in having the flexibility and capabilities to quickly and efficiently serve all these important areas. To discuss your specific application interests, or to explore setting up a trial evaluation of Biolog's phenotype microarray technology, or to place an order, please contact the following departments.